Um, so today I'm going to talk about creating at-home meaningful microbiology exercises for students. So I have been doing at-home food microbiology labs since 2018. And we kind of started doing this out of necessity. There was not time available in the food labs on campus, so the students couldn't really actually make edible food anywhere on campus. So we had them do the processing at home and bring subsamples into the lab to actually analyze and do quality assurance on. But it turned out to actually be a good thing. And I think the students are actually learning more and enjoying the course more, doing different reactions in their homes. So over the last couple of years, I've been kind of making different iterations of the course. We've done both cream cheese and camembert cheese, talking about food preservation. We've tried both wine and hard apple cider to talk about process control. And in this one, if they don't have good process control, they get apple cider vinegar, which is also yummy. But it's an excellent learning opportunity for them to learn about how quick microbiological processes can get out of control. We've been doing sauerkraut or yogurt and yogurt to learn about species succession. Kombucha and kefir are great for teaching about multi-species communities, and they both tend to work out very well. And we've been doing antibiotic resistance bacteria in food. So today I'm going to talk about sauerkraut and yogurt, but if anyone has any questions about the other ones or how to adjust them to have the students do them at home, I'm more than willing to share both lab exercises and advice. So the yogurt lab is very easy for students to do at home. You can purchase all the materials you need on Amazon, so there's no science-y purchases that need to be made. You can do the entire thing for about $22. All you really need is a thermometer, a starter culture, and pH paper. But if there is budgetary allowance, the yogurt maker does make it work a little bit better. The students have to provide some things from their own home, like a sauce pot, a whisk, some milk, and yogurt, but it's actually a very easy lab. The only thing that we did learn here is that there's about 100 different combinations of different bacteria in starter culture that are all available from Amazon. So one thing that I've learned is to get the simplest one, the Lactobacillus bulgaricus and Streptococcus thermophilus. If you start with some of the more probiotic blends of yogurt, it doesn't work as well, and it's very hard to track the actual microbial succession in what's going on there. Whereas if you start with just two, you can actually see through some of the results how nicely the ferment is progressing. So in this particular lab, all they have to do is basically boil milk or warm milk till it reaches a temperature where the bacteria are happy, 35 or 40. Add in the starter culture, mix it up very nicely, and then put the mix in the yogurt maker or just keep it in a warm place. It works fairly well either way. They leave it to incubate 13 hours, and they can monitor the pH over the course of this 13 hours just using the pH paper. And they can also monitor the texture and consistency of the yogurt by like moving it around with a spoon or kind of adjusting it that way. So this is actually kind of a neat lab in that there's a couple of food microbiology things coming and happening, and there's also a couple of species things happening, and you can really address this in class and you can really make them think about it to get the right answers as they're writing up their discussion. So for yogurt, optimal water retention is 4.2 to 4.6. And if they let the reaction go to 24 hours, what they're going to see is that the whey from the milk starts coming off and the yogurt becomes too thick and you don't get the nice yogurty consistency. So then you can talk about what happened there in terms of the milk fat excluding water at a certain acid point. So at the start of a yogurt fermentation, most yogurt starters have approximately equal numbers of strep and lactobacillus. And you can see this if you plate them out, or in this case, if you can't plate them out this year, you could give them fake plates to count for this. And you'll generally see these two, I'll talk about it on the next slide, but there's a very clear growth pattern for each one of them. So in a yogurt fermentation, they start, strep and lactobacillus start at the same concentrations and they end at the same concentrations. But strep has a biphasic growth pattern where at the start it can grow more quickly than the lactobacillus. But as it's growing at these numbers, it's lowering the pH. And L. bulgaricus actually grows better at a lower pH than S. thermophilus. So it uses a lot of the byproducts from S. thermophilus to quickly grow, and then it levels off. As it's leveling off, a lot of the byproducts from the L. bulgaricus growth stimulate S. thermophilus growth, and it then grows more rapidly to end it the same way. If you have a technique that's available to your undergrad lab, like 16S or sequencing of some sort, you can actually watch this happen in real time. It's harder if you just have the media, because there's not a great media to differentiate the two. So in regular years, what I've had students do is take a sample of their milk after inoculation of their cultures at 0 hours, and then they take a sample of their yogurt at 13 hours, they keep it on ice, they bring it to the lab, and they plate it out on HHD agar. Now this is kind of neat, because when you plate these samples on this, homofermentative lactic acid bacteria form nice green colonies with yellow sheens in behind, whereas the heterofermentative form white colonies. So it's easy to differentiate the two. Like I said, there's not a good media to differentiate these two because they're both homofermentative. So basically what you see is an increase in colonies between 0 and 13 hours. But I'll get into some interesting activities for that later. So we can't do this obviously this year because we don't have on-campus labs. So there are software where you can make theoretical plates for students to count. So this year, after they conduct their lab, I'll be giving them these theoretical pictures of plates to count of what their results would have been like. And they can still do the calculations and figure out growth curves and overlay their growth curves with their pH to figure out everything that was happening in the milk during the reaction. Now in 2018, in this lab, we always compare the products we make to something you can buy commercially at the grocery store. And when we plated out our yogurt from 2018 that we bought at the grocery store, a lot of it was sterile, which was a big surprise. But the other big surprise was that at the colonies at the end, we didn't see the homofermentative. We saw more heterofermentative popping up from the commercial yogurt than we saw from our own yogurt. And a lot of the students were confused and looked into this over time. But what we eventually figured out was happening is this is the probiotic cultures that are added in to a lot of the commercial yogurts. So the bifidobacteria show up as white colonies on the HHD agar. So I did keep this for this year, and we'll see without the lab time if they can figure out what the differences are between the commercial and the yogurt they make in their own home, but we are keeping it for this year. So it gives them a lot to think about. So the consistency of the milk is happening, and it's extending shelf life because of the lactic acid production. So as an assignment for them to do as well as this, I want them to look at the grocery store when they go for their weekly trip and make the purchases that they need for their lab each week to compare the best before data of 3 milk products and 3 yogurt products to actually see this in, you know, an industry setting where you are getting the shelf life extension. And it's also interesting to see if anyone will come up with the differences between the homemade yogurt and the commercial yogurt and the actual right explanation for it. The homemade yogurt will also have a very different consistency than the commercial yogurt. And this is actually because of the add-in of gelatin in commercially prepared yogurt, but this is not maybe applicable to regular microbiology courses and more of a food science question. So every year I've taught this course, and this might be because I'm in an agricultural faculty, but students insist on using their own raw milk to make yogurt and cheese. So it requires kind of a weekly encouragement not to use raw milk to make these products for this class. So if you're at an agricultural facility, it's something to consider, because it's definitely something I didn't foresee happening. But if you're not, maybe you won't have that problem. Discuss not eating anything. Although these are fairly easy ferments, we only get about a 50% success rate, even with yogurt and sauerkraut, really easy things to make. So it's really worth having a slide deck of common mistakes or common gone wrong fermentations to show them really what they shouldn't be eating. In addition, when we do this on a regular year, I usually circulate in the lab fairly often, smell things, look at things, visual examinations, and look at their data and discuss signs of something they shouldn't be eating and discuss what probably went wrong. So on these years, I'm fairly comfortable with students eating what they made if it's clearly safe. But this year, I'll be recommending that no one eat anything they make for this lab, just because there's the added layer of looking at their data to add safety to it. I've also noticed that group members, everyone, at least in my classes, are very keen, and everyone contributes to the work, but everyone in this class seems to contribute unevenly to, do they participate in the actual hands-on stuff, or do they do the write-up? And you usually get one or two very keen students in the class who do all of the ferments for the group. I don't see anything wrong with that, but it does seem to go that way more often than not. A good follow-up lab for this after the sauerkraut is actually to do, after the yogurt is actually to do sauerkraut. And this is a little bit more of a complicated succession. You're not adding a starter culture. You're relying on the microbes that are naturally on cabbage to do the fermentation. So in this instance, you have 3 different species of bacteria that participate in the fermentation. This lab can be done for under $5 a student. They basically need half a cabbage, a couple tablespoons of salt, and an airlock. And what you see is that when you mix all these things according to a standard sauerkraut recipe, you get the heterofermentative bacteria growing faster at the beginning, lowering the pH very, very rapidly, but it stops growing after the acidity gets to about 2% because it's the heterofermentative. Homofermentative bacteria are more acid tolerant, so they come alive at about day 3, but grow more slowly. And then at the end, you get a different homofermentative bacteria to come up. So if students make the sauerkraut over a 21-day period and collect a sample every 2 to 3 days, you do actually see on the HHD agar the succession from heterofermentative to homofermentative. And it actually does work out very nicely in the lab. So we will be making sauerkraut again this year. They're getting the plates that I've made just in their lab books to count, but they will be able to overlay the different species with the pH that they're collecting on a daily basis. So I think it'll still turn out fairly nice. I was encouraged not to do this when I started doing it in the lab, but after a couple years, the student comments, they've been very encouraging. So one thing that I think will go very well this year is students actually really do like to do experiments in their homes based on the last couple of years of my experience. And I kind of just wanted to share some of their comments as a way of encouraging people that the students actually appreciate doing this as opposed to not liking doing things in their own homes. So they do seem to really enjoy that. The other thing that I've noticed over the last couple of years is that when undergraduates come into my lab to start graduate degrees, they have really, really sloppy, like at best, notebook taking abilities. So I've put this into this course so that we're looking at their notebooks, we're giving comments on how to improve note taking, and we're actually giving them grades for it. And I was really upset that we were going to lose this aspect of the course this year, because there's not a really good way to sit down with students all over the world and look at their notebook taking and help them address it. Then I came across ELNs, and I spent about a week this summer going over approximately 10 ELNs for which ones were the best ones. And we ended up choosing to use OneNote for this course. There's some nice articles about different labs that use OneNote as their standard lab notebooks in research labs. But there's also some about using it for a lab notebook in undergraduate labs. So I've shared those links there. But we set it up for this course for the fall. And it's very nice because you can share the actual lab book you can share the lab manual within the lab notebook. So they have all of the labs they'll be doing this semester. Each student has their own lab book, which I and the TAs can mark or help or assist with at any point during the semester. And the students can actually take their own notes, add their own pictures, link to their own larger data sets. So I think it'll actually be a very nice way of doing this. And I may not go back to the paper notebooks, seeing how easy the electronic lab books are. So I think I'm out of time, and that is it. Thank you.
Thank you, Jen. And that was a great presentation. I learned a lot. I will moderate the questions. If you have a question, you can type it either in the chat, or if you want to ask it, please feel free to unmute yourself. And if you like, share your video and we can have a conversation. We'll take a few questions. While we wait, I can ask a question. So you said that you used a software to create the pictures of the plates, and so is that software free, or is that something that's proprietary available to you?
I use BioRender, so that addition is.
Yeah.
Thanks.
So yeah, BioRender, I just learned about that recently. A lot of students have been using that. Great. So that's really cool. With the electronic notebooks, OneNote is part of the 365 suite. Is that right? Or
it is part of the 365 suite.
Oh, great. Laura has a question. I will let you ask your question, Laura.
Hi, thanks, Jennifer. That was really interesting. I teach a third-year microbial ecology and diversity course, and we make sauerkraut in class. And not in lab, but just as a demo in class, and then we have sausages and sauerkraut later. And I've never asked permission for that, but having everything go through our safety office now, our safety office, it's really difficult to get permission to do anything that involves the students potentially eating anything or hurting themselves. And so like even just like the knife for the cabbage for the sauerkraut may be more than my university is willing to sign off on. And so I'm just curious if you've had any experience with getting these various protocols set up, especially since like explicitly yogurt seems like a thing everyone would just like sneak a spoonful of.
Yeah. So they are being told not to eat the food this year. I understand that some of them may sneak a spoonful of, but there's not a lot I don't think there's a lot I can do about that. In terms of safety, the protocol at our university was any at-home activities had to be categorized as low risk, mid risk, high risk, and low risk, you had to agree on it with your chair and proceed. Medium risk, you had to get the permission of the Dean of Students to continue on with. So all of the activities in this were classified as low risk.
Great. Thank you, Jen. So Jen has kindly agreed to share all of the materials she talked about today. If you would like those materials, please either send an email to myself or CSM, and they can forward it back to us, and we will share with you the folder on Google Drives that you can use for your purpose. If you have any other questions, please feel free to ask them at the end of the, keep adding them into the chat, and we will get to them at the end. I suppose I'm the next speaker, so I suppose I can go get started. Share my screen. So I'll just introduce myself. I am Maria Davis. I am a laboratory instructor at the University of Regina. I teach here in the biology department, and I teach labs on plant physiology and genetics. I teach both 2 levels of genetics, 200 and 300 level labs. And today I'm going to talk to you about sparking student interest in primary research through proposal writing. And this is an assessment, it's a semester-long assessment that I do in the 300 level genetics course. And before I get started into the details of this assessment, it's a research proposal, I wanted to go into a few reasons of why I do this. The original assessment that was in this course was a lab report. So the genetics course that we teach here, I co-teach it, and in the lecture, students generally focus on bacterial genetics with a little bit of focus on viral genetics. And in the lab, I give them hands-on experience on some bioinformatics, some molecular cloning, and then we cap off the semester with the proof of concept with CRISPR-Cas, where students design a tracer RNA to deactivate GFP. And so they get to nicely first make bacteria glow and then get them to stop glowing. So in a lab report, which was the traditional assessment at the end of this course, the students are performing a lab that has been designed by the instructors and then using the prescribed maybe 5 or 6 references that are provided by the instructor. It's very rare that students go outside and find their own references. And so when we think about a lab report, they're simply describing the experiment that they performed, predicting what they expect to have as results and discussing those, and then interpreting some of that data based on the prescribed references that have been provided. So when we overlay the Bloom's taxonomy on a traditional lab report, the students maybe would be just, in my opinion, hitting the lower levels of the Bloom's taxonomy in this example of this course. And since they're in the 3rd year of their undergraduate career, I wanted students, and thinking about my experience, I wanted students to be really thinking about or hitting the higher levels of Bloom's taxonomy, of creating their own research question, which would maybe get them to invest a little more and dig a little deeper into molecular genetics. So that came. Thinking about this, I came up with a very I came up with a very vague set of instructions. The instructions are not this vague. This is not the only thing that the students see, but the main idea is the students want to write a research proposal about a gene, any gene that they are interested in, because we're in a genetics course. This word limit has increased. I was originally inspired by the NSERC graduate proposal that students have to write. I wrote in my 1st year of my master's, which is 1 page with tiny margins, and trying to get as much information into that was difficult. And I have, since I started this, slowly increased the word limit that I provide students with, so that they can really focus in and do their research and share what they're excited about. So I just want to quickly talk about the learning objectives and lay them over the Bloom's taxonomy. The students are told to keep a general audience in mind, but specifically someone that's taken the course that they are in, so that they don't have to repeat or explain too many things. To keep that word limit short. The main learning objective is to generate a research question in molecular genetics, which really hits all of the levels of Bloom's taxonomy, where they need to remember and recall the concepts that they've learned in class, be able to understand and apply them to be able to generate a good research question and create a proposal that can answer that question. The rest of the objectives also, as you can see, do generally hit the higher levels of Bloom's taxonomy. So I've been doing this assessment over the entire semester for 2 years now. I started this in 2018, and in 2018, I wanted the students to start writing or get a feel for writing. So I started with some mini writing assessments, mainly because I remember when I started writing in graduate school, which is the first time I really started writing, it was a very daunting task. And so I started with these assessments, and during the same time, I asked students to meet with me informally to sort of form up their proposal in their mind and as an outline. We had about 32 students this year in 2018, so it was not too bad to get that those meetings done and formally over the entire month. In the middle of the semester, we did an open peer review, and this was done using a discussion forum, and only on the introduction section in this case. This worked really well that it was open. Students were very good at giving feedback and really kind in a really kind way. The students also got feedback from the instructor on the entire report, and then finally their final draft was due at the end of the semester. I honed this a bit more in 2019, where I scrapped the mini writing assessments and just asked them to write up the whole draft of the entire proposal, a 2-page proposal at the time, and the feedback was given as requested in this case. This didn't work as well as I had hoped because, as you can imagine, it was just the usual suspects who were at my office all the time getting the feedback, while a lot of others didn't really either plan properly to get feedback, to have enough time to get the feedback, or simply were too shy or nervous to ask. This draft was then peer reviewed, and the peer review was graded. So the draft wasn't graded at all by me. They just got feedback as requested. Each student got at least 2 other peers reviewing their assessment, and they were asked again to get instructor feedback, and as they requested, I would give them feedback. I did this because it was a much busier semester that year, and we had increased number of students from 32 in 2018 to about 41 in 2019. And at the end of the semester, they submitted their final version of their proposal, and I added a new activity last year, which was the funding forum. And in this activity, students were broken up into groups of 5 and given 3 proposals to either fund or not fund. Turned out to be a very exciting activity for them. They did enjoy it, but I did do it as a bonus to check how good the students would participate, what participation would be in it. And I found that most students were really excited about doing this funding forum from their feedback. So this coming fall, starting next week, is going to be the 3rd iteration of this assessment. And again, I've changed it quite a bit. In this iteration, it will be a group project because we have shot up in the enrollments. We have about a 50% increase in the class size. So now we are at 60 students this year. And for this year, I have added a individual pitch that they will start the semester with, and this can be a video pitch or not, just a written pitch. This pitch will be graded, and as a group, they will pitch it to each other over in a breakout room on Zoom and pick their gene that they want to go forwards with. They will then together create a draft, each student focusing on one of the sections, 3 major sections of the proposal. And this draft will be graded so that they will get instructor feedback from me. And finally, they will create their final submission. They can, if they choose to, because this is a bonus, create a video group pitch where they all create their final pitch on why they want to study this gene or why this gene is exciting. And then finally, they go into a virtual funding forum. The final draft is due about a week and a half before the funding forum. So this will give me some time to create groups for the funding forum and then distribute the proposals to those groups so that they can read it prior to the forum, and then over Zoom in breakout groups discuss these. They also have an assessment due from the funding forum. They have to write a short report, which is worth a small amount of 2% of their final grade. So they will have to submit that as well. So anything that you see here in bold was a bonus assessment, and all of the grading was done using rubrics. The rubrics are really text heavy, so I'm not going to show them to you, but I would be happy to send them along. So I have rubrics for not only the proposal, but also for the peer review as well. So I started this talk saying that I really wanted to create this proposal because I wanted to give students more experience of really feeling or having a personal connection to their course. For whatever reason, they were interested in the gene. So I asked them, this was just done last year, if they preferred the final assessment to be a lab report instead of the research proposal, because at the start of the semester, I heard a lot of grumblings about, oh, I have to do so much work. But it turns out of the 50% that did respond, majority of them, a large majority of them, really did appreciate having this research proposal instead of just a lab report as an assessment. And before I leave you, I'm just going to give you two titles that the students of two of the projects that were submitted. And in the first one, the student wants to sequence the unknown region of a enzyme, polygalacturonidase, which is an enzyme in tomatoes, and it is what causes tomatoes to get soft. So they were hoping to understand how to prevent this and increase the shelf life of tomatoes. And in the second example here, the student was interested in increasing the acid and ethanol tolerance of yeast that are specifically used for brewing sour beers. And they plan to do so, or they suggested they would do so using, by incorporating two genes from bacteria that are known to be acid tolerant and overexpressing them in this yeast strain. Again, thank you for listening. My contact information is right here. Feel free to contact me either via Twitter or my email, and I would be happy to pass on these resources to you. So, yeah. So if you have any questions either for myself or for Jen, I would be happy to pass those on.
Maria, I've got a question. This is a really interesting assignment. I love that idea. When you were talking about sort of this grumbling that the students had, thinking about, oh, this is going to be so much work, that was kind of the first thing that came to my mind, but it's nice to see that they, in the end, really enjoyed it. I guess what I'm wondering is, when you were coming up with this assignment idea, were you basing it on something you'd seen before, or was this something that just sort of came out of a need that you saw and a gap in your own kind of experiences as an undergrad?
I suppose I think I've seen this since that I have seen people do this, and I don't believe that I was the first one to invent this, but I did come up with it because of a need. I get a lot of pre-med students in the course, and I remember being one of them and also thinking I loved it because I didn't have to write anything, but then I was sorely disappointed as soon as I hit grad school that I had to write so much. So that was one of the reasons that I really, people's ideas of what they want to do with their life change, like mine did. And written communication as well as oral communication, because I'm trying to get them to do more video pitches and more science communication assignments in class as well. And so, yes, it came from basically the fact that I had so much trouble getting started to write, that I created that. Thanks for the question.
Thanks.
Anya.
So enjoyed both of the presentations. Maria, I was curious about how you graded the peer feedback, the peer evaluations.
The peer evaluations were also graded using a rubric, and the rubric includes, I can just share my screen very quickly. Do I have it on here? Share. And this doesn't show the entire, oops, I'm sorry. Oops. It doesn't show the entire rubric, but I give them instructions on what to write, and I've told them that how many words they need to write, and they always have to start with their understanding of what was proposed. And then there's, I think this is just a short piece of it, but there's four criteria and 4 categories to assess them in. And of course, I'm acknowledging one of my colleagues from the University of New Brunswick who I spoke with a lot about peer review, and he's done peer review, and he has used, he came up with the questions of what students need to think about and reflect on when creating a peer review. So this is how I use this rubric to grade it. Thank you.
That's great. I've done a really simple peer review evaluation, but it wasn't nearly this in depth, and I think this would be a lot more helpful when you're trying to get people to give good peer review. So, thank you for sharing that.
Hi Maria, I have a question if that's okay. I really like your idea of pick your favorite gene, and because it personalizes it and makes it more kind of interesting for the students. I was wondering though, what sort of range of feasibility did you get among the genes that the students were picking? Like, were they easy to work with, and was it like, when they went to the end, was it a good learning experience for them?
That's a great question, actually. It was, I think, a great learning experience for them and for me, because most of them are, as I said, pre-med students. So they were picking human genes, and I'm a microbiologist. So working with them, and I tell students to come see me. And so we had a lot of informal meetings, and working with them, for them to understand that this is a process and not everybody knows everything, I think was really great. There was one student that chose a gene from whales, I believe, because the whale genome had just been sequenced, and she'd read something that said that this gene is probably what is involved in the longevity of whales. So the idea was to somehow study that gene, clone it, and try to put it into humans. And when I heard that, I was like, that is really ambitious. I don't know. But it turned out the number, she got really into it. And turned out the number of exons and how large the gene was, she wasn't really thinking about how she would clone it. And she ended up throwing that one aside and going to a new project, but she learned a lot about just genes and considerations that you think about when writing a proposal.
Cool, excellent, thank you.
No problem. Well, if there is no other questions, I will ask you to please sign up for the next session that we have. We have our very own Tanya, who will be presenting, Dr. Noel, and which will be followed by a informal discussion that will be led by Dr. Rubin on the committee. And thank you for coming again. And if there's nothing else, we will see you next week.